Hello everyone, welcome to today's video, and today I will be explaining salt. Salt, it's not sodium chloride that I'm going to explain. I'm going to explain skin associated lymphoid tissue. Okay, so we'll start with the first thing, which is the most important thing, which is epidermis. We all know what is epidermis, right? So let me increase the size. Okay, so this is our epidermis. Let me place it right over here. Now in epidermis, what happens is sometimes what you have is sometimes what you get is lot of antigens. So these are these structural representations of antigens that are coming from outside, and they are trying to enter inside of our body. So they have to come through the epidermis, which is the outermost layer of our skin. Right, so role of epidermis immunity is it acts as a sensory layer. It's very important to understand that it is the topmost layer, and it is also having the sensory functions. And there are so many things that are present in epidermis. Let me use this area a little bit. Okay, so the antigen is trying to penetrate inside, and for some reason, maybe there is injury in the skin, there is some damage, the antigen is able to enter inside the body. Antigen can be any foreign particle, antigen can be any proteinaceous substance, antigen can be coming from the pathogen itself, it's wire, anything, right? So it will encounter it will be entering through epidermis, and it will encounter some of the important things that are there in our epidermis. The first important part is keratocytes. So there are keratocytes that are present. I'm going to use this cellular representation to show that these are the let's assume these are the keratocytes, and they are present right in the epidermis. And the function of keratocytes, they are immune active structural cells. Almost 90% of the epidermal cells they are made up of keratocytes, and they are surprisingly, these are sophisticated immune cells. They recognize pathogens, antigens by using toll-like receptors. They can release cytokines like interleukin-1, TNF-alpha, IL-6, all those things these keratocytes can do. Right, and as you can see this in this image, that keratocytes are present in the background, and they are creating the immune environment of the epidermis. Okay, so now next cell, which is Langerhans cells, and you also have you can see the illustration right over here. So Langerhans cells are also another type of cells. They are basically nothing but dendritic cells. Okay, dendritic cells of the skin are the Langerhans cells, and role of dendritic cells is specialized antigen representing cells. They what they will do is they will interact with the antigen that is coming. They will interact, and they will try to maybe phagocytize the antigen, interact with the antigen, try to break it down. They will process antigens internally, right? So if that is the bigger part of the cell I'm going to show you. So these antigens that are there, they are going to come inside of this cell, and they are going to get processed, and they will be represented on the surface of these kind of cells, so that other cells, they can get activated. Right, so as you can see, the processing of the antigen will be happening inside these vesicles, inside these cells. So these are Langerhans cells. Basically, if you can see the antigen presenting cells of the skin, and they are dendritic cells, and that is why you see those dendrites coming from these Langerhans cells, right? So these are some of the important parts of our epidermis. Now there are another things that are called intraepidermal lymphocytes. So I have illustration for lymphocytes right over here. These are our lymphocytes. If I elaborate it, it looks like this. Okay, so these are lymphocytes, and these lymphocytes, they are nothing but they are our T cells. Right, so these are the cells that are residing inside the epidermis, and their role is to basically take care of the infected cells, try to activate B cells. They are important in viral infections, they are important in immune tolerance. So many important functions are there. You can have memory lymphocytes also, you can have helper lymphocytes also, you can have cytotoxic lymphocytes also, although I'll be explaining them in the later part of this illustration. Right, okay. So now let's move on, and this happens inside. Everything is happening in the epidermis. So you can imagine how dynamic is your epidermis. It's not static; it's dynamic. After that, what you have is dermis. So let me bring a dermis right over here, And I'm taking dermis from right over here, as you can see. So this is dermis. Okay, let's fill it out. Okay, fill it out like this. This is dermis, and this is the second part of the skin after epidermis. So the second region, this is dermis. And what happens in dermis, that we need to understand. Okay, so in dermis, what you have is one important component, which is lymphocytes are there. But in addition to lymphocytes, you have these interesting cells. These are macrophages. Macrophages are present everywhere, and the role of macrophages. Their role is to phagocytize those foreign invaders like bacteria, dead cells, debris. Their role is also to present antigens to other immune cells. Their role is to also release cytokines like pro-inflammatory cytokines, so that inflammation can be triggered, and because of that inflammation, you can have other cells, other immune cells, they can be brought to this particular region. So let me also represent the release of inflammatory cytokines right over here. So this is our macrophage which is releasing a lot of, let's create more. This is our illustration, right? So we can make it more amazing by showing a lot of inflammatory response. Let's imagine that you have very strong attack of the bacterial cells that is going on right over here. Your Langerhans cells, they are getting activated. Macrophages, they are getting activated, and they are releasing a lot of cytokines right over here. Right. In addition to that, you also have lymphocytes. So I'll put some of the lymphocytes right over here. And in addition to lymphocyte, you also have the entry of your entire lymph vessel, which is coming from here. Okay, so let me move it a little bit on that side. So let me show you right over here a lymph vessel. So this is the lymph vessel, and I'm going to bring the lymph vessel right over here, and I have to increase the size of this lymph vessel so that it's visible to you. So it starts from dermis. So now you have a better understanding of lymph vessel. So it will be starting from here, and we'll have, I just created the white part right over here because I wanted it to blend with the lymph node, right? So this is the lymph node region coming right after the dermis, and it will look like this. Okay, so that it blends with the environment, so it will look like this. So this is the lymph node region, and then you have lymph vessel, which is getting connected with the lymph node, right? So that's the lymph node, and this is the inflammation that is causing a lot of issues. And inside the lymph node, you have a lot of amazing things that are going on. So Langerhans cells, they are going to enter from here. Let me show you the movement of Langerhans cells, right? Entering inside the lymph vessel. Right, and inside that, what is going to have is antigen presentation. So this is antigen presentation going on inside the lymph node right over here. So you can see, let me zoom in. Okay, so you can see the antigen, that the squiggly line that you're seeing is red line that you're seeing is the antigen part that is getting processed, and some of the part of that particular antigen is being shown to lymphocytes right over here. So that is the process we called antigen presentation. So the part of the pathogen is getting processed inside the cell, and that is getting presented to the immune cells. That happens inside the lymph node right over here. Interesting, right? So all these pathogens, they are getting engulfed by these immune cells, and then inside that, activation of other immune cells that is happening. So antigen presentation is happening inside this lymph node. All right, and after there is this recognition of this particular pathogen, the antigen presenting cells is going to present this to the immune cells, and then TH cells, they are going to get activated. This is our TH cell right over here. Okay, so this TH cell, T helper cell, got activated, a little bit grayish in color. This is bluish in color, right? You can see. Then TH cell, they are going to release a lot of cytokines. Okay, so there will be the release of cytokines. Let me show you with that arrow. Okay, so then you have the release of cytokines, and these are our cytokines again, right? These right over here, that's the cytokines, and cytokines, they got released, and these cytokines, they're going to activate further the B cells. These are our B cells right over here. B cells, now B cells further is going to convert itself into plasma cells. That's the antibody secreting cell, which is the plasma cell, and that contains a lot of endoplasmic reticulum like this, as you can see over here. Let me make it a little bit bigger. You have a lot of endoplasmic reticulum, and it's a protein manufacturing facility. It's going to produce a lot of antibodies. Now, antibodies are right over here. These are our antibodies. So antibodies, they are going to get secreted from these antibody-producing plasma cells, and you're going to have a lot of antibodies, right? A lot of antibodies are going to be there in the circulation. And this is again our adaptive immune system. So in the lymph node, what is happening? A lot of antibodies are getting produced, and these antibodies ultimately are going to block these the pathogens that are there in our skin. So pathogen can penetrate in the epidermis, they can penetrate inside the dermis, but these antibodies, they are going to be beneficial in blocking the pathogen or the antigen. And because of this, then we will be able to block the infection. So in dermal region, you can have lymphocytes, you can have CD4 helper cells, you can have CD8 T cells. These are cytotoxic T cells. You can have memory cells. What I've shown you right over here, I'm talking about these lymphocytes. So in dermis as well as in epidermis, you can have these lymphocytes, and they can be helper cells, they can be cytotoxic cells. The difference between helper and cytotoxic cell is one is going to help. So as you can see over here, this is the helper cell. It's going to help release cytokines. It's going to release the cytokines, and then it's going to help in activation of the B cell. And further, B cell will be converted, and I think it's appropriate if I use arrow. B cell is going to convert it into plasma cell, and plasma cell is antibody secreting cell. So it is releasing a lot of antibodies, and these antibodies, they are going to further neutralize our pathogen, right? So this is the most important part, and there is one thing that I've not explained in detail, which is lymph node, and where the adaptive immune system that is showing its true nature inside the lymph node. That is the region where all the soldiers are there, and where actual training is happening. And what happens? You have T helper cells. As I've explained to you, they are going to activate the antibody secreting cells. They will produce the antibodies that are highly specific to those antigens, right? And then the entire system is going to function. This entire system and all these components, they are salt. This is salt, skin-associated lymphoid tissue. It's really important for the protection. It's really important for neutralizing the invading, pathogens. And there are these important other components like continuous immune surveillance happens because of the salt. Rapid local defense, long-term immunological memory is there because of the salt, and immune tolerance against harmless antigen is also one of the important aspect of salt. If we talk about clinical relevance, it works when there is skin infection, when there is allergy, when there is autoimmune skin disease, when you have vaccine delivery. All those important aspects are there when it comes to clinical manifestations or clinical relevance of the salt system. I hope with this video, I think you will have a great understanding of salt, and I'm sure that the way we explain in Biology Gold, we try to explain every topic in a very, very interesting and illustrative way, and that helps the student in understanding the concept really well. For that, we just seek your support for our channel, because if you support us, if you subscribe to the channel, if you comment on the video, if you share the videos, it helps us a lot. It makes us motivated so that we can create more useful videos like this, and so that you can understand complex topics in a very, very interesting way, right? So this is very, very unique to Biology Gold. We are the one only creating these kind of videos, so stay tuned and watch all those videos. Let me tell you another thing. If you are interested in knowing about any specific topic, just comment on this particular video, and I'll try to make that one for you, so that your knowledge, your understanding, your learning never stops. All right, I want to show you something. This is the resource that I've created. It's called Research Goal, where I create a lot of different courses. Depending upon the need of the students, you can have multiple different courses in various different domains, and they are all high quality. They're going to help you in your research. That is why it's called Research Goal. There are multiple courses that are available. Let me show you these courses. As you can see, you have courses related to PyMol also. If you want to learn PyMol effectively, you have protein analysis and editing course. If you want to do molecular docking, which is really important for drug discovery, this is the course that is available. If you want to do some data analysis, you have data analysis for biotechnologists, Python programming if you want to learn. And all those examples are not computer based examinations or computer based curriculum. They are more catered towards biology or cell biology, microbiology, biotechnology based programming examples. So it's very, very easy for you to understand how you can learn programming. You have machine learning in cancer biology, you have R programming foundation course, you also have review paper course which can help you write review articles. And in the long run, I want to create the amazing resource for research students who are interested in doing research. Everything I want to provide, all the experience that I have, I will be putting inside this particular website, and you can just take these courses, all those videos, they are available. And another thing, the videos inside these courses are also, many of the videos, they are also freely available, right? If I show you, if you go to curriculum, you can see many of these videos, they are also free for watch. That means you can complete all these 4 videos and you can understand the basic knowledge about the course. And if you are interested and you think that, okay, I should do this particular course, then you can complete this entire course and you will be getting a particular certificate of completion once you complete the course and also answer the quiz. There are a lot of resources that I'm providing along with the video. Also, there are notes, there are scripts that I provide that helps you in understanding the topic really well. All right, thank you so much for seeing this part of the video and understanding the research goal. Please, in the description you can find the link for the research goal. Please go and explore that particular resource. All right, let's continue our video.